Hi, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are joining today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. Many of you may have attended ASPB's annual plant biology meeting in the past. Each year, this event brings together plant biologists to talk about latest research topics. In 2020, this event was held completely online and had over 2,500 participants. ASPB's program committee is currently working on making plans for next year's event. The overall theme for this year is plants powering the bioeconomy. If you have ideas for workshops and would like to submit your ideas, please send a proposal by December 4th. Although we are currently planning for a face-to-face meeting in Pittsburgh, organizers should describe how they would convert their workshop to a hybrid or virtual conference format if that becomes necessary. I'd also like to encourage you all to apply for a travel award to attend Plant Biology 2021. More information about funding opportunities can be found on our website. Notice that the first two deadlines are early in December, which is coming up soon. Today's talk is the 20th talk in our Plantae Presents webinar series. If you haven't already done so, check out our archive to watch all the previous talks in the series. You can find information about each event on our blog and subscribe to our YouTube channel. If you're interested in presenting in this webinar series, consider submitting a 7-minute video of your research to one of our upcoming, and they've both been really fun. In this format, we choose the top 4 videos and show them to our audience. Then we have the speakers join in a panel discussion at the end. It's been a really cool way to include more people and have more interactive webinars. So I hope that you are able to submit your research topics here. If you're having trouble connecting or need to leave the webinar early, please know that a recording of this webinar will be made available along with all of the associated materials within a few days. Today's talks will each be 20 minutes each. As you listen to each talk, add your questions for our panelists in the Zoom question and answer Q&A section. Questions that are specifically for our panelists should be shared within the Q&A section, just to make them easier for our moderator to navigate. You'll also notice that there is a chat area open. This section is for you to introduce yourself, say where you're from, and connect with your colleague. My amazing colleague, Mary Williams, has been adding useful links for you here as well. All right. Elisa Delaglio is with us today to moderate our discussion. Elisa is a postdoctoral research associate in INRA, the National Institute of Research Agronomy, and holds a PhD in plant biochemistry from the University of Grenoble in France. Her main interests are plant enzyme modulation and plant stress responses to biotic and abiotic stresses. She is a member of the board of Aricera, the Italian International Researchers Association, and she's also an ASPB Plant Physiology Assistant Features Editor. Thank you for joining us today, Elisa. I'm going to turn it over to you now to moderate the discussion.
Thank you, Katie. It is a very great pleasure to be here today. We are actually living a chaotic moment now, which includes a global pandemic and a very important elections going on at the same moment. So I think it is even more important to abandon the news for a little while and remind ourselves that there is much more in the world than that we are not alone in this planet, but we share it with many other living beings, such as plants and insects and so on. And all of them are also fundamental to preserve ecosystem and agriculture, and therefore our future. So with no further hesitation, I will now introduce our today's first speaker, who is Gustavo MacIntosh, who will talk about colonization strategies and resistance responses in soybean-soybean aphid interactions. He joined Iowa State University in August 2003, and he is now professor in the Roy J. Carver Department of Biochemistry, Biophysics, and Molecular Biology. Gustavo has a biology BS/MS from Universidad Nacional de Mar del Plata and a biochemistry PhD from Universidad de Buenos Aires in Argentina. He was a postdoctoral associate at the DOE-Plant-Research Laboratory at Michigan State University, and then a research scientist at the University of Delaware before joining Iowa State University. He is also committed to increasing the participation of underserved groups in science and to make ASPB a place where everybody is accepted and supported. So thanks, Gustavo. I can let you now share your screen and see you later for the questions.
Right. Thank you for the nice introduction. And thanks also to Mary for the invitation, all of you for joining me. I hope you can see my slides now. So my lab is interested in understanding aphids.
Slides, actually, sorry.
Yeah, we don't actually see them yet. Yep.
Can you see them? How about now?
Now we see it.
Yeah. All right. Okay, the title is gone.
From the top.
So as I was saying, aphids are really cool because they are specialized insects. They can feed from the phloem sap. And so they have this modified mouth part that is basically like a little straw inside of this mouth, and that allows them to penetrate different tissues on the plant until they reach the vasculature and they start feeding from the phloem. As they do that, they secrete salivary secretions that allow them to penetrate different tissues, and also they have proteins that control different plant responses. At the same time, they produce little damage, but the plant recognizes its damage and also recognizes some of the proteins that allows the plant now to mount the response. They are also parthenogenetic, at least in part of their annual cycle. And so that means that they reproduce really quickly. They have the ability to evolve quickly because of that. And they can become indifferent to insecticides, for example, resistant to insecticide really quickly. And so they become really bad problems for a lot of different crops around the world. There isn't as much study on plant aphid interactions as there is in other systems in plant pathology, but the interaction between the plants and the aphids has been modeled based on what we know for plant pathogen interactions. And so we have the equivalent to PTI, which is herbivore trigger immunity. The plant recognizes herbivore associated molecular patterns and trigger immune response, basal defenses. Insects that are specialized has effectors in the saliva that block the signaling for this basal immunity. And so these plants are that are susceptible to aphid colonization now have what is normally associated as the effector trigger susceptibility. So these plants are losing. But there are plants that have evolved the ability to recognize those effectors, and those plants have resistant genes that had proteins that recognize the effectors and trigger an alternative pathway for cellular signaling that triggers now a defense response that shares a lot with PTI, but is stronger and is faster, basically. And so in this case, the plant becomes resistant to infestation by the aphids, and the plant's winning. So this model came out about 10 years ago by Hohenhauer and Boss. And it was at the time I was starting with this project in my lab, which started as a side project, but it helped me contextualize the things that we were trying to do. I'm in Iowa, and so I had decided to work in soybean because I wanted to do something relevant to the state. And here is either soybean or corn. And so the model that I was working with was soybean and the soybean aphid. We knew very little about this system because the soybean aphid had just been introduced in the US. And so my approach was pretty simple. Luckily, we have soybean germplasm that was susceptible or resistant to the aphid. So we have a nice system to work with. And basically, we just look at gene expression analysis within metabolic profiling. And then from that, we synthesized the information we had to generate hypotheses that we could test. And one example of that is a paper we just presented a little while ago. And in this case, what we did was to keep aphids on the plant for a very long time. It's actually 3 weeks with aphid infestation on the plants. We knew that in the compatible interaction, if we put aphids, we see a suppression of defenses early on, but we wanted to know what happens late during the interaction. And what we found is that eventually the plant started defending itself, and we saw all markers in this transcriptome analysis, all the markers of classical PTI, and we think that chitin may be a hump. But the interesting thing here is that among the things that we saw induced in response to aphid was the branch of the phenylpropanoid pathway that is responsible for the production of isoflavones. And so we wanted to know if that was really leading to accumulation of isoflavones. We measured isoflavone production in these plants, and we found that in response to this long aphid infestation, we had increase in all the isoflavones we could detect. And we were really interested because isoflavones are normally characterized as antimicrobial compounds. They have been very well studied in that sense, but we didn't know anything about their role in the interaction with insects. So the first things we wanted to do was know where in the plant are those isoflavones accumulating. So for that, we did this mass spectrometry imaging in which we can generate a 2D map of the different chemicals that accumulate on a leaf. And this is what you see here. You can see here, this is just an imprinting of the leaf to give you an idea where the vascular tissue is. In the control plants, we didn't have any accumulation. And in the aphid-infested plants, you can see accumulation here in green or yellows of the different isoflavones that I'm showing the channels here. So 3 different isoflavones that I can show. And you can see that instead of being in the vascular tissue where the aphids are feeding, the accumulation happened in the mesophyll near the vascular tissue. So what we think is happening is as the aphid is penetrating the tissues to reach the phloem, it has to taste the cells as it's moving along and is finding this chemical, which suggested to us that it could be a deterrent, so bad taste in the leaves that could deter the aphids from feeding. So an undergrad in the lab, Chiavarri, decided to do this choice experiment in which she put some leaves on a solution that was just a buffer, and then other leaves on a solution that have dicing, this one over here, and then put aphids in the middle and let them pick. She came back a few hours later and counted how many aphids had gone to either side, and she found, after several repeats of this, a clear preference of the aphids for the plants that didn't have dicing in the solution. So we think that the isoflavones are really working as deterrents for aphid feeding. So going back to that model, then we think that we have evidence for these PTI-like responses in the aphids, but we were also interested, as I said, in what happened early on when there is suppression of defenses. And again, from a very early transcriptome analysis that we did in the lab, published a long time ago now, a different student, Matt Studham, decided to check what happened early during the 1 day after aphid infestation of 7 days. And he designed this analysis in which he looked at biosynthetic genes for the different phytohormones. And he also looked at marker genes for this hormone. So whether the plant is making a hormone and whether it's responding to it. And he found something really interesting. So if we look at jasmonic acid, at day 1, he saw an increase in the biosynthetic genes for jasmonic acid. And then if you look at the response, he saw a very nice response. But at day 7, he saw a much larger increase in the biosynthetic gene expression and 0 response. So clearly the aphid is doing something to the plant that doesn't allow it to respond to JA anymore. At the same time, he saw a big increase in ABA, both synthesis and response. And that was interesting to him because JA is associated with effective defenses against insects, while ABA is normally associated with abiotic stress. So his hypothesis was that as the aphid reached the plant tried to defend itself using a JA-dependent response, but the aphid is able to induce a decoy that is ABA-dependent, and that is antagonistic to the defenses. So then he graduated, so he couldn't test this, and a different student, Charles Canobi, decided to test this. And so he did a simple experiment again. He put aphids on the plants and left them for 7 days. And then he had control plants without aphids, of course. And they treated the plants with jasmonic acid and waited 6 hours and looked at the jasmonic acid response just by looking at gene expression of a classical marker for JA responses, the PIN2. And what he found was that in the controls without aphids, he had a very nice increase in PIN2 expression after jasmonic acid treatment. But this response is very attenuated if the plant had been colonized by aphids for 7 days. A different student, Jessica Hohenstein, decided to check now the role of ABA in this effect. And so she did an experiment where she looked at what happened, the same experiment that Charles had done, but now in plants that were defective for ABA. And in this case, she got these plants by using virus-induced silencing, targeting a biosynthetic gene in the ABA pathway and a transcription factor that controls ABA responses. When she did the same experiments as Charles, she used mechanical wounding in this case, but it's the same idea. And she saw the same attenuation of the wounding response when the aphids are present on the plant. But then in the ABA defective plants, this attenuation due to aphid colonization is completely lost. You can see here that it doesn't matter if the plant was just wounded or if we had aphids and then wounded, we have the same response. But what's more interesting is that if we compare these plants that are the normal plants, let's say, versus the ABA defective plants, we see that even without aphids, so the green bars here, we see a big increase in response to wounding. So what we think is happening is in soybean, the plant uses ABA as a way to modulate the intensity of the jasmonic acid response. And the aphid is exploiting this normal mechanism by increasing the ABA response in the plant and then blocking the ability of the plant to induce a jasmonic acid response. So that means that the aphid can block jasmonic acid responses, but is that important for the aphid? So Jessica decided to look at how aphids do in those ABA defective plants. And for that, she just put aphids on the plants, came back a week later and counted how many aphids she had. And this is the controls. And these are the ABA deficient plants. And you can see that the deficient plants have fewer aphids. So if the plant cannot make ABA, they become more resistant to aphids, which put another way means that the aphids need ABA for successful colonization. Charles had measured jasmonic acid accumulation, and he found that in response to aphids for 7 days, after 7-day colonization, there is a big increase in jasmonic acid. But so Jessica thought that, okay, if the plant is making jasmonic acid, maybe what's happening is there is no conversion from jasmonic acid to the active form that is jasmonic acid isoleucine. She checked the accumulation of J-isoleucine, and she found that there is no change if the plant had or don't have aphids. So what we think is happening is somehow ABA is blocking the conversion of JA to jasmonic acid isoleucine. So we know now that aphids can block the defense responses in a compatible interaction. And we also wanted to know what happened is that an incompatible interaction. How does the plant defend itself? And as I mentioned, in this germplasm, the soybean germplasm, we have a lot of different resistant genes, about 12, I think, have been characterized. And I work with RAG1 and RAG2. And the idea is that RAG1, the product of RAG1 will recognize an effector and trigger the defense response, and RAG2 will recognize a different effector probably and trigger a defense response. So the question that we had was, is the immune response triggered by RAG1 the same as the one triggered by RAG2? And this question comes from experiments have done by breeders, where breeders use this tool, gene pyramiding, as a common tool for making better crops, right? And the idea of gene pyramiding is combining two different resistant genes into the same genotype. And what people have seen, and we saw in our plants too, is that if you combine two resistant genes, RAG1 and RAG2, you can see here aphid performance in a susceptible plant versus the monogenic resistant lines. And then in a combination of the two, you see that the combined line has a stronger resistant phenotype than either one of these two lines. So Martha Natukunda and other students in the lab have the hypothesis that The reason why the pyramid is more resistant is because RAG1 and RAG2 induce different subsets of the defense response. So in transcriptome terms, what she was expecting is that RAG1 will induce some changes in the transcriptome in response to aphids. RAG2 will induce different changes in the transcriptome, maybe some overlap. But in the pyramid, what she will see was an addition of the RAG1 and the RAG2 responses. So this additive effect will explain why the RAG1, RAG2 pyramid had a stronger resistant phenotype. So that was very easy to test. She just analyzed the transcriptome responses for susceptible plants and the 3 different resistant lines. And we did it at 6 and 12 hours because we knew already from previous experiments that the resistant response is established really quickly. And when she looked at the overlap, we saw something really interesting. First, she looked at what things are common, and she found that there are some genes that are common to all the responses, including the susceptible plants. But at 6 hours, this is a small number of genes. 100 genes is about 10% of any of the resistant responses that are common to all the plants. At 12 hours, that increased to 600 and something genes. So it's about 50% of any of the responses. As you can see here, all of the responses seem to catch up. So what we think is at 6 hours, we have very different response, but at 12 hours, the responses seem to converge. But the important thing is to look at the overlap between RAG1 and RAG2 that she was looking at. And so when we look at the genes that are differentially expressed only in one genotype, that's the big surprise. She was expecting to see in the pyramid basically a combination of this response and this response. But instead, what she found is that the pyramid have a unique response. And the largest set of genes is the ones that are only differentially expressed in the pyramid. So she, instead of an additive effect that she was expecting, she found this synergistic effect. Combining RAG1 and RAG2 doesn't give us RAG1 plus RAG2, it gives us a completely different response. How do we explain this completely different response that likely is responsible for this increased resistant phenotype? We think it has to do with some sort of transcriptional factor interactions, because we still have a RAG1 signaling and a RAG2 signaling that have to act to induce something here. So we think that maybe combination of transcription factors that are unique in this pyramid could explain this result. So to understand that, we did a transcription factor network analysis. We actually had to do the analysis by mapping everything on the Arabidopsis genome, because the interactome with Arabidopsis is much better than what we have for soybean. But we got a very nice interaction network for all the transcription factors that are expressed in the pyramid. And then we looked at, I'll show you here one zoom to explain what we did is we looked for each Arabidopsis gene, we have several homologs sometimes in soybean. And so what you see here in this particular hub that we thought was very interesting is we have the MIP15 Arabidopsis gene has 2 homologs that we saw differentially expressed in our soybean data set in the pyramid. And in red, I'm showing you one homolog that is induced in the RAG2 response and in the pyramid. And in blue, one that is induced in the RAG1 response and in the pyramid. So you can see here signaling from RAG1, signaling from RAG2, and then interacting in this hub. Here in WRKI46 is actually a gene that is differentially expressed in the RAG1 response, in the RAG2 response, and in the pyramid. And here we have some other combinations that suggest signaling from the individual RAG genes that converge in this hub. The interesting thing is that around those, we see a lot of yellow transcription factors. Those are only differentially expressed in the pyramid. So what we think is that this hub allowed the conversion of RAG1 and RAG2 signals, and then this is amplified by interactions with these other transcription factors and lead to the expression of synergistic genes. So put it in a more graphical way, we think the RAG1 will induce a transcription factor that's not enough for inducing expression of the gene. RAG2, and this is a different one, is still not enough. And then RAG1, RAG2 has both signals from RAC1 and RAC2, the two transcription factors, and that induces the defense gene. You could also model it's different. It could be that because those transcription factor normally works at heterodimers or homodimers, and we have homologs that are very similar, you could have some different complex combinations that induce this expression. So that's how we think is working in our system. Now the question is, what are those 1,000 genes doing? Why is this plant more resistant? Well, we did several different analyses. I don't have time to show you, but as a summary of all those analyses we did, we think that after recognition of the aphid effectors by RAC1 and RAC2, there is induction of transcription factors in this MIB-Wurtke hub. We know that these two MIBs in soybean induce isoflavone production, individually induce isoflavone production. So here we have combination of two, we think even a stronger response. We know that MIB15s and Wurtke 33 in other plants increase lignin content, so secondary cell wall biosynthesis. A different hub that I didn't have time to show you is repressed, and we know that this GNC transcription factor controls photosynthesis, so it's a positive regulator of photosynthesis, and we found a repression of photosynthesis in our plants. And we know that Wurtke 40, which is up here, is a negative regulator of photosynthesis, so we think that this decrease in photosynthesis in the plant. Decoupling of the electron transport chains because these photosynthetic genes are repressed leads to the production of reactive oxygen species that have been studied pretty well in other ETI responses. And ROS, together with the increase in lignin, will contribute to cell wall modifications that increase physical barriers in this plant. And at the same time, ROS induces HR-like responses that are also being associated with some resistance to aphid responses. And some people have suggested that photosynthesis decrease will also limit the photoassimilator viability, but we don't have real evidence for that. So in summary, I showed you that in compatible interactions, the soybean aphid can block jasmonic acid defenses, but if you leave the aphids for a long time, the plants start fighting back. We think that ABA blocks this jasmonic acid signaling by inhibiting the activation of JA to JA isoleucine. We saw an increased resistance in the pyramiding of RAC1 and RAC2, and we think that's due to the particular combination of transcription factors that allow the synergistic response to occur, which is very early, happens around 6 hours, and that involves production of chemical, physical defenses, and repression of photosynthesis. And we also found that isoflavones function as aphid deterrents. And I already showed you the people that actually did the work, but we also have a lot of help from other people that were very essential for our work, and this is funded. And with that, I'll stop. Thank you, everybody, and I'm happy to answer any questions.
Thank you, Gustavo. There are already various questions for you. So the first question is, do we know anything about jasmonic acid ABA interaction under aphid infestation in Arabidopsis or any other plant? Or do we know any other process in which ABA influences JA response?
Yes, and actually the the right response will be, it depends. So JA have different effects, this JA and the JA ethylene pathway. And we think that this effect on the jasmonic acid responses that we are seeing is more in the JA ethylene pathway than in the JA pathway alone. But it also depends on the plant and the type of interactions that you are studying. What is the effect of the ABA on JA? So, and Arabidopsis is a little more complicated to compare Arabidopsis to soybean. So in Arabidopsis, the JA salicylic acid antagonism has been very well characterized. But in soybean, we are seeing that JA and salicylic acid seem to work in a cooperative way in the interaction, in the defense against aphids. So I'm very cautious when I compare with Arabidopsis. On the other hand, we have a paper published a few years back where we also show that for Arabidopsis mysus persicae, if we have ABA deficient plants, the aphids do worse. So the ABA deficient plants are also more resistant to mysus persicae in the Arabidopsis mice interaction. So we think that there is this approach of blocking defense responses using ABA, also in that system. What the target is may be different.
Okay. There's another question that could be maybe correlated. Since salicylic acid also initiates from shikimic acid pathway common with phenylpropanoic pathway, but you saw changes only in isoflavone and lignin and not in salicylic acid. Can you comment on that?
Well, we don't know, and the 3 weeks in infestation, we did see a very strong induction of the isoflavone pathway. We also see induction of the early part of the pathway, but we didn't measure salicylic acid in that particular interaction. If you were back to my slide where I showed the 1 and 7 day transcriptome analysis, we actually do see an increase in salicylic acid early on at day 1, and we also see a repression of salicylic acid responses at day 7 when we see the repression of JA responses, but we haven't measured salicylic acid in our plants.
And besides isoflavones, what other secondary metabolites have you studied or do you think may be important in the defense response?
The main phytoalexins in soybean are isoflavones. So we have looked more to isoflavones than anything else. We haven't really focused on any other phytoalexin. We have looked at lipids, we're working on cuticle, we look at cell wall in terms of metabolomes, but we have not looked at other phytoalexins yet.
Last question, I think. Do you think that MIB15S and WILQ33 are using defense hormones such as jasmonic acid to overcome growth via growth hormones?
Yes, I think that jasmonic acid is involved in this response. And it's not just from our experiment. We do have a little bit of experiments on that area, but the paper just came out showing that JA increases the resistant response in the resistant plants. And so we think JA is involved. I also think that salicylic acid is part of that defense response too. We don't see a particularly different set of phytohormone signaling in the pyramid compared to the other resistant varieties. So we think that JA and SA participate in all the 3 resistant genotypes we saw. But I don't think that's what's make the difference. I think the difference is probably the ability of the pyramid to induce this HR-like response very quickly, because we see suppression of photosynthesis much earlier in this pyramid than in the other genotypes.
And about the 2 mutants, RAG1 and RAG2, do you know which are the genes that are mutated in these 2 phenotypes?
Those are not mutants, those are resistant genes, right? Yeah. So what are the genes? We don't know. The 2 genes were fine mapped, I don't know how long ago, maybe 8 years ago. And the problem is that, like always, the reference genotype doesn't have the genes. And so you then need to go to a resistant plant and try to find it. It turns out those regions are always amplified in the resistant plants. There are MBS LRR genes in those regions, and both for RAG1 and RAG2, there is 1 or 2 MBS LRRs that are the candidate genes, but the people that worked on those have not done the experiments to show that they are yet. And I don't want to step on other people's research, so we are not doing that. But we'll keep waiting for a while.
Of course. Thank you very much again.
Thank you.
So now I think we can move on to our second speaker for today. So the second speaker is Jyothi Vaddasery, who will talk about calcium-mediated defense against insect herbivores. Jyothi is a scientist at the National Institute of Plant Genome Research. She obtained her MSc in genetics from Indian Agricultural Research Institute in Delhi, in India. She earned her PhD at the Friedrich Schiller University in Jena, Germany, as an international Max Planck Research School Fellow, and studied the role of calcium signaling in perception of endophyte Piriformospora indica in Arabidopsis. She then pursued postdoctoral work at the Cornell University to study temperature regulation of plant defense. Further, she moved to Max Planck Institute of Chemical Ecology in Jena and worked on the role of calcium sensors in plant defense against Spodoptera herbivory. Her group at National Institute of Plant Genome Research works on early signaling pathways involved in plant defense against insect herbivores, role of calcium channels in the process, and the regulation of jasmonate pathway by calcium. Thanks you, Jyothi. You can start.
So thank you for the kind introduction, and I want to thank ASPB for inviting me to give this talk. I just want to check if the slides are moving. Yeah. So my talk today will be about calcium-mediated defense against insect herbivores. This is the insect that we are working on, Spodoptera litura, and we work on Arabidopsis in our lab. So as you know, Spodoptera is a very destructive generalist insect pest. It's a lepidopteran insect, which is a very voracious and fast feeder. So if you have a very hungry caterpillar, it can finish a plant very quick. And it feeds on aerial plant parts and with very wide host range. And recently you would have heard of the fall armyworm, which is a related species, Spodoptera frugiperda, which is causing a lot of damage in maize. So it's a very important insect pest which we are studying, and it's a chewing insect. Unlike Gustavo's talk where you heard about aphids, this talk is about lepidopteran chewing insects. So when we talk about plant defense, so in my talk, I'll introduce a little bit about the, before I come to our work. So the plant, what are plant defense strategies against insects? So when you talk about insect feeding, there are two things that the plant perceive. The plant perceives wounding, which you can see here, and it also perceives the oral secretion in the saliva, and which activates a very rapid signaling via calcium, which I will explain you today. We know that jasmonic acid signaling pathway is the key pathway which is involved in 90% of all the responses against lepidopteran insects, which in turn leads to direct defense mechanisms like chemical defenses, secondary metabolite production, for example, or the glucosinolate production in Arabidopsis, or other defensive proteins and protease inhibitor production, so which makes the plant less tastier to the insects. So these are basically the direct defense mechanism. There is also very active indirect defense mechanism in plants where they release volatiles, and which in turn attracts predators or parasitoid to eat on these insects. So this is what we have known from a very long time, and many labs have been working on it. Now, when we talk about early perception, it's very important to understand what do plants perceive upon insect herbivory. So plants perceive two different signals, kinds of signals. One is a non-self signal, or the signals coming from the insects. They can be HAMs, herbivore-associated molecular patterns, or the recently discovered egg-associated molecular patterns, elicitors, which are basically released from the, which come through the saliva of the insect, or there can be effectors which kind of try to downregulate the plant defense. So these are the exogenous signals that you see. There are also endogenous signals, that is, the plant is also able to perceive its self-injury. And these are the damp molecules which you have often heard of, for example, the ATPEPs, the ATP, which is released out of the cells, many other compounds which are released, which tells the plant in, as a self signal that you are wounded and you need to activate an immune response. So when we talk about herbivory, it is a lot of signals which come together, and probably many receptors which work in tandem. Many years ago, while doing my postdoc, we realized that if you take oral secretions, this is the Spodoptera, you tap its head, you get these oral secretions. We asked what happens when you add this oral secretion on a plant. So we use this calcium reporter. There are many transgenic calcium reporters which are available in Arabidopsis, and in this experiment we use the ecorine. So this is what happens if you put water onto an ecorine leaf disc. There is no signal, but if you put this oral secretion from the insect onto the calcium reporter, you'd get a very nice elevation of calcium, which is a very fast signal. So we know that calcium is one of the first response of a plant to oral secretions from the insect. Over the years, this field has developed a lot, and now we know that it is not only calcium. When we talk about the fast response of plant to herbivory, because insects are feeding so fast, and so there are many other components of this fast response. And one of them is the electrical signal, which has been discovered from the lab of Ted Farmer, pioneering work from his lab, which shows that there are electrical signals which travel from local to systemic leaves and activate jasmonic acid pathway. We know that from our work that there are calcium elevation involved. We are talking about signals which occur in a matter of a minute. And we know that much downstream, there is the jasmonic acid pathway, which is leading to the other secondary metabolite production. So this is what happens in a local leaf. What I mean by a local leaf is a leaf which is bitten by an insect. But it's not only that leaf, but also systemic leaf, which shows a fast response to herbivory. Many years ago, after this paper came from Ted Farmer's lab, that the glutamate receptors are involved in systemic electrical signals. So there is systemic electrical signals and GLRs are involved. We try to look whether there are systemic calcium signals as well in a collaboration with Edgar's lab in Halle. And what we found here, this is a control plant. These are calcium reporters, which we image, echorin imaging. What you see here, you're in a control plant where there is no insect, you don't particularly see any signal. But here, this is a plant which in which the insect was trapped on the leaf 8. And as it feeds, you can see a higher calcium, not only in the fed leaf, but also in the systemic leaves. So we now know that there are systemic calcium and electrical signals. Now, with the advent of modern methods to image calcium, we have much more powerful methods to image calcium. And as you might have seen with the GCAM, this is an imaging with the calcium reporter GCAM. And you can see when we cut the leaf here, which we call the local leaf, you can see a very fast systemic signal which goes on to the, these vascularly connected leaves. So the plants respond very rapidly to wounding and herbivory, and also warn the unwounded leaves that you're attacked and you have to mount a defense. So these are these discoveries are what shape our research questions, and there are 3 major research areas in our field. One is to identify the calcium channels which are involved in herbivory perception. We know that there is a big calcium elevation occurring when insect is feeding. There are a whole lot of dams and hams and many receptors. And if there has to be a calcium elevation occurring, there has to be also a multitude of calcium channels which let this calcium in for the signal that you see. We also know that there is a jasmonic acid pathway activation, and we are also interested in this pathway of how calcium regulates it. So the first question in our lab is, what are those insect responsive early signaling calcium channels and their function, and how does direct and indirect herbivory defenses influence them? The second question is, are there herbivore signals which are activating or blocking these calcium elevations? Because the insects would have also evolved to overcome such a defense. And this is one thing we are trying to understand. And the third topic is about jasmonic acid isoleucine induced calcium elevation, and if we are looking for novel receptors and channels which might be involved in this process. So today my talk would be about calcium channels in herbivory perception, the first topic, and it is based on this published work last year, which came last year. So we are, as I told you, we are looking at calcium channels which are involved in herbivory perception. As you saw from that last video, that there is a very huge surge of calcium when a plant is wounded. So what happens in the cell? How are these elevations produced? So calcium elevations are usually produced due to opening of ion channels, which are present in either in the plasma membrane or in the tonoplast. They can be in different places in the cell. So this is our influx channels, or what lets the calcium inside the cell. Yeah. So we know from the work of Ted Farmer that GLR 3.3 and 3.6, for example, are also such a family of calcium channel which are involved in systemic electrical calcium and jasmonate response. And so the GLR 3.3 and 3.6 have very different also cellular localization. So one is in the ER and another is in the vacuole. So we so far do not know of any CNGC channels which are, or any channels which are located on the plasma membrane and are involved in this calcium elevation that you see. So when I started my lab around 6 years ago, my postdoc, Mukesh, started this work by doing a microarray. And we found that there are a lot of CNGC channels, which if you treat the plant with the Spodoptera oral secretions, after 30 minutes, you see a lot of CNGCs getting upregulated. We decided to do the functional characterization of one of them, which is the CNGC 19. So the CNGCs are calcium permeable channels. They have 6 transmembrane domains and an N and C terminal. The C terminal is very special that it has a cyclic AMP or cyclic GMP binding domain, which is called the CNVD domain, and a CAM binding domain. So the calmodulins basically regulate the opening and closing of the channel, and also the cyclic AMP. So the CNGC 19 is a member of a group 4A of this family, and the 19 and the 20. And the group 4B is the very famous CNGCs, the CNGC 2 and 4, which are involved in many other innate immune response against microbes. So when we got this result, we went further and we did our gene expression studies and found that it is induced upon both insect feeding, very high fold of upregulation and also very rapidly. And it is also in wound and oral secretion induced, that is, it is both wound and enhanced by OS application in Arabidopsis leaves. And we also found that it is not only in the local leaves, that is the leaf that is wounded, but the CNGC expression is also pretty high in the systemic leaves. And we decided now to further look at their functional role in herbivory. So we had these tDNA knockouts of the CNGC, the 19-2 and the 19-1 lines. If you grow them normally, they do not have any phenotype. This is the kind of plants we use for our herbivory experiments. And then in these plants, we put young insects and allow them to feed for 8 days. And then we look, how does the phenotype look like? So this is in the wild type, and this is in the 2 mutants, where you see that the insect has eaten more. We call this an insect weight gain assay, and we measure the weight of the insects which are feeding on these mutant lines. So this is in the wild type, and you can see in both the mutants, the insects are feeding more. The COI 1-16 is a positive control as a JA receptor. So the CNGC 19 loss of function results in increased Spodoptera litura feeding and a decreased defense. So this is what we got from this essay. So this got us really interested. And we started to look, where is the CNGC 19 expressed in Planta. So we fused this, the promoter to GUS, and we found that it is mostly localized to the vacuoles, as you can see here. And upon wounding, you can vasculature, sorry. And as after wounding, it spreads into primary, secondary, and tertiary veins. So it's a vasculature, leaf vasculature localized gene. And we also found the GUS signals at the base of trichomes here. Yeah, this got us thinking, and so it was postulated that CNGC 19 is a calcium channel. Nobody had proved till this point that it is really a bona fide calcium permeable channel. We collaborated with a group in Bangalore and with Professor Matthew's group, and we went on to look if this is really a calcium channel. So we expressed the CNGC in Xenopus oocyte. And we did this electrophysiology experiment. So if you give, and as I told you, this is a CNGC channel. So the cyclic AMP acts as a ligand here. So if you give only dibutyryl cyclic AMP, you do not see any inward activating any currents here. If you give cyclic AMP plus sodium or potassium, also you do not see any activity. But with calcium, you see a very strong inward rectifying current. And this indicates that it is a hyperpolarization activated channel. And you can see that sensitivity with different concentration of calcium, and that this is really looking like more of a calcium, more calcium permeable channel. Yeah, so this got us very excited, and we also had data which showed that this is plasma membrane localized. So we went further, and my PhD student Ram asked, what happens if you do not have this channel? What happens to the calcium signal propagation? So what you see in this video is a single leaf, and we are making a wound, simulating herbivory. We make a wound in the midvein, and you can see that these are the calcium signal which spreads in the plant as you make a wound. Yeah, you can see this signals are spreading through the vasculature, and then they are entering into the intervascular spaces. Yeah, so a rather fast signal. Now we took this GCAM in the back, crossed it to the CNGC19 mutants, and you can see what happens in a mutant. So we did similar experiment. We cut near the midvein, and you can see how the signal proceeds in such a leaf. So in the mutants, the signal seems to have lost its way through the vasculature. So it is very aberrant in its spread. It is extremely slow when you compare it to the wild type signal, and it is attenuated in its response. So in the CNGC19 mutant, the calcium signal propagation through the vasculature is simply not happening in the way it should happen in a wild type. We quantified this response, and this is in a wild type. You see a very fast calcium response in a local leaf, whereas in 90 we see a very reduced calcium response. And this proved to us that if you do not have this channel, the propagation of calcium in the local leaf is inhibited. So we haven't really looked up at the systemic leaves, and this is something that we are doing. Further, we found that this expression of CNGC19 is also regulated by certain receptors. So the PPR1 and 2 are DAMP receptors. They are receptors of ATPEPs. And when we took the PPR double mutant and looked up at the expression of CNGCs, so these white bars are wild type. So when you wound it, you see a CNGC expression, and with OS it's enhanced. But in the PPR mutants, if you do not have these receptors, you see very less expression of CNGC19, implying that these receptors are regulating these channels. We also looked whether these channels are impaired also in the sensing of ATPEP, and surprisingly, we also found that the ATPEP perception is also reduced in these mutants. The black bar is the wild type. So they seem to be involved in ATPEP perception and regulated by PPRs. We still do not know how this is occurring. Then we went downstream. So the PEPs, PPR, everything you saw was very early response to herbivory. Then we looked up what happens to the jasmonates or the secondary metabolites in the whole process. And this is the jasmonic acid content in the wild type and the mutant. And as you can see here, both the JA and the JA isoleucine levels are highly reduced in the mutants, and also the expression of all the JA marker genes, telling us that the insect is feeding more on these plants also because the jasmonic acid levels are highly decreased here. We further went on and asked ourselves what are the effects on the secondary metabolite level, and for Arabidopsis it's mainly glucosinolate, which is a readout for herbivory response and anti-herbivory response. They are amino acid derived secondary metabolites, and the breakdown products are very toxic to Spodoptera. So then we asked, we measured the levels of total glucosinolate in wild type and both the mutant line. So surprisingly, we found that both the mutant line have very low levels of glucosinolate, total glucosinolate, and which is due to reduction in aliphatic glucosinolate. So the indole glucosinolate levels are relatively unchanged. Yeah. Upon feeding also, this is in constitutive level without any insect, and upon feeding also these levels are very low, and this implies that the glucosinolate levels are really less in these plants, at least the aliphatic. Then we asked ourselves, why is it that the aliphatic glucosinolate levels are less? So we did an untargeted metabolite profiling in these lines, and we discovered that these lines are somehow over-accumulating methionine. So both CNGC19-1 and 19-2 seem to accumulate methionine and its transport form as methyl methionine. Yeah. So if you look up at the glucosinolate pathway, this results hold importance because methionine is the precursor of aliphatic glucosinolate pathway. And if you remember, our aliphatic glucosinolate levels are down in the mutants, whereas the methionine levels are high. So we looked up at all the genes in the pathway in both the mutants and tried to look if anything changes in these lines. And we found that the BCAT4, a specific gene which is involved in conversion of methionine to 2-keto acid, is downregulated in these mutants. We further went on to look profile all the BCATs in this mutant, and we found that apart from BCAT4, there is also 1 and 2 which was downregulated, but which are not involved in aliphatic glucosinolate production. So we believe that this downregulation of aliphatic glucosinolate is due to the effect of BCAT4. So come to the end of our hypothetical model based on the single CNGC that we have. So when an insect bites on a plant, it releases dams and effectors, everything. And there are a whole lot of receptors which are present on the plasma membrane, many of which we do not know of. But what we know that PPR is also a major player here. And it activates CNGC, so not CNGC alone, because you do not see a complete loss of signal. There could be other CNGCs probably on the membrane which act together and form a tetramer. We do not know, but this CNGC mediated, this channel opens and you see a calcium elevation. We have also shown in the paper that the CAM2 is involved in this interaction. And this calcium elevation is required for jasmonic acid and aliphatic glucosinolate production, which ultimately leads to plant defense against herbivory. So this is the publication from which this model is. We have also shown this year that the CNGCs are not only involved in herbivory defense, but they are also involved in basal immunity against plant microbe interactions. So in our future work, we are trying to understand how herbivory and CNGC channels identify the molecular switch that either opens or closes or opens the channel, how they are gated by calmodulins, and as seen from many studies, so this is the latest study which shows the role of CNGC20 in cell death and how phosphorylation of this channel is important for to inhibit cell death. And in this, it is shown that the BAC1 phosphorylation actually is responsible for the CNGC 1920 degradation. And we believe in herbivory also a receptor or a receptor complex proteins might be involved in phosphorylation and activation of these channels. We are also looking at functional analysis of other CNGC channels and its role with respect to crosstalk with GLR, ROS, and jasmonate. So a lot of postdocs who are working on all of these projects. Another important aspect that we are looking is how do insects, avoid such a calcium elevation. So we recently have published a paper where we have tried to, predict the effectors from the salivary glands of insect. So these are 3 major salivary glands. And one of our very interesting candidate is the calcium binding protein isoform from the insect salivary gland. So we are testing the hypothesis whether these insect effectors can stop this short and long distance calcium signaling that you see here. And we know then Mahindra we're working on this project along with Paramita. So apart from that, as I told you before, we have also projects where we are working on jasmonic acid isoleucine induced calcium elevation and a forward genetic screen, trying to identify look for mutants which do not have this JA isoleucine induced calcium elevation, and trying to look for novel receptors or channels which are involved in sensing JA layers dam. Yeah. So with this, I would like to thank all of you. This is our beautiful institute in Delhi. So we are a premier plant science institute in India. We have 30 plant biologists working in this institute in various areas of plant biology. Do check in our website and to know about our work. And I would like to thank my group for the amazing work that they've done in the last 6 years. Our collaborators, Professor Matthew and Kirti from NCBS, Axel and Michelle from Max Planck in Vienna, and Dr. Arokisami from ICGP. So our lab is funded by DBT, our funding agency, ICR, Max Planck Society through the Max Planck India Partner Group, and M. Thank you.
Thank you, Jyothi, for this nice presentation. I think we have time for at least a couple of questions. So the first question is, if you tried to treat the CNGC19 mutants with Spodoptera oral secretions, and did you see a difference in the calcium response?
Yes, so we did treat the CNGC mutants with the oral secretion of the insect, and we found a reduced calcium elevation with this OS, yes.
Okay. And did you look specifically to which vascular tissue CNGC19 is expressed?
No, but previously Petra Dietrich lab has already shown that CNGC19 is phloem expressed, but we really haven't looked in much detail. But this is already known that it's a phloem expressed, like almost like GLR 3.3.
Okay. The last question, I think, is, would it be right to assume that CNG19 works in allowing calcium influx inside vascular cells from other neighboring cells?
Yeah, so the CNGC19 is not only acting in the vascular cells, it's just that you see the vascular signal first when you image. So the spread to the intervascular spaces is relatively slow because I think it occurs through the plasmodiesel matter. But yeah, the CNGC works, at least it's one of the channel components which works in letting in the calcium. Because it's such a, there are many channels involved in this process. You don't, as I told in the talk, you don't really see a loss of signal. It's just that the signal propagation is slower and lesser. So yes, it's definitely one of the channels which are involved in the process.
I just saw that actually Gustavo couldn't use the Q&R, so he put a question in the chat. Do you know if any CDPK is involved in the transduction of calcium signaling?
With Spodoptera. So yeah, the CDPKs seem to be in our system. We don't work on CDPKs, but we know that CDPKs in general are able to phosphorylate CNGC. So CNGC channels might also be activated as a result of an initial calcium elevation by some other channels, you know, and the CDPKs may be involved in phosphorylating CNGCs and, you know, self-propagating this wave. Yeah, so CDPKs are definitely involved in the CNGC regulation. Yes.
I will try to resume 3 questions in one to end everything. So did you look at any specific compounds in insect or secretions, and are there differences in various Spodoptera species concerning that? Do you expect variations if we look at various species of Spodoptera?
Oral secretion is a very complex mixture. It has a lot of plant fragments in it. And apart from that, a lot of insect salivary gland proteins. So we are trying to look at it one by one, trying to look which are these proteins. We really do not have, you know, one elicitor which we can tell, yes, this is what is involved. And this really, working with insects is really tough because of this fact that there are no elicitors which we can, for example, the fatty acid amino acid conjugates, which are elicitors in other crop plants, are not working in Arabidopsis. So this also, we are also trying to look for, you know, those specific compounds in the oral secretion, which is very complex at the moment.
Okay, thank you very much. I think I asked all the questions that were in the chat, but if I forgot, you can also have a look and maybe answer specifically to each one, but I think we are good. Thank you very much again.
Thank you. Thank you
for the great moderation, and also thank you, Jyothi, for staying up so late. I guess it's 10 p.m. there now, so we really appreciate you taking the time to stay up. Thank you also, Gustavo. That was both excellent talks, and we hope to see you at our next Plantae Presents event. For now, we'll see you later. Thank you, everyone.
Bye. Thanks, everyone.